Okay, welcome everyone. This is the worksheet that we started, or this is a picture we started drawing last week, Wednesday or Thursday. These are chromosomes. These are a homologous pair of chromosomes. These homologous pairs are chromosome number 1, pair number 1, or number 2, or number 3. We have 23 pairs of chromosomes in our body cells. These are homologous. This is one of the pairs. Where did one of these chromosomes come from? Your mom, and then if the one came from your mom, where'd the other one come from? Your dad. That's where the genetic information from these chromosomes came from. We have 23 pairs. We have 46 total chromosomes in our body cells. And almost all cells in our body, let me rephrase that. Almost all cells in you are called body cells. There are very few that are not body cells, and those we call sex cells, and those sex cells are egg and sperm cells. These homologous pairs have bands on them called genes. These genes code for a particular trait, like hair color, eye color. If we oversimplify, we could say your height, genetic information. They code for particular things. So that's what that yellow stripe represents, is a gene. That's what that black stripes represent, is a gene. It's a gene, it's a location of genetic information that codes for a particular trait. Within these chromosomes, they have identical genes. So if that yellow band is hair color, the one on the left was from mom, the one on the right, the yellow band, is also a hair color gene. This came from dad. Now, does your mom give you, is it possible that your mom passes on the same hair color trait as your dad? Mm-hmm. Yes, it's possible. Is it possible that your dad and mom pass on different hair color traits to you? Mm-hmm. Yes, it's possible, right? It depends upon what their genetic information is. So when we talk about genes are the traits, it's the location on the chromosome, it's something like hair color. The types of hair color, or the variations of the hair colors, we refer to as an allele. An allele is a form of a gene, and the example of an allele would be like blonde hair, or black hair, or brown hair, or red hair. Those are different alleles, those are different variations of the gene that we call hair color. Okay, so a gene is a location for a particular trait. An allele is a form of that gene. It's possible that your mom passes on the same allele as your dad, but it's also possible that they pass on different alleles. Their alleles may be the same. In the homologous chromosomes, the alleles may be the same. However, they may be different. The alleles might be the same, however, they might be different. But in sister chromatids, what's true about sister chromatids' genetic information? They're identical. They're genetically identical. So that means that sister chromatids will have the same allele because they're genetically identical, and they will have the same gene as well. Sister chromatids, a gene, allele, chromosomes, sister chromatids. We missing anything? We're good. Oh, diploid, haploid. Right? We wanted to define that.
Did we do that on this?
I think we did.
I think so.
Yeah, right there. Yep. Okay, so we wanted to find two terms before we kind of move on. There's two terms that we're going to kind of introduce. We're going to use today a little bit, but we'll get more into using the terminology along the way. There's two terms. One's called diploid and one's called haploid. And sometimes when we abbreviate diploid, we use the letters 2N, whereas haploid we represent as 1N. And when we talk about a diploid cell, we talk about cells that are diploid. A diploid cell is a cell that contains both sets. Make sure I get the wording. Both sets of the homologous chromosomes. They get both sets of homologous chromosomes. They get a set from mom and they get a chromosome from mom and dad. Both are present. Okay, a diploid cell is a cell that contains both sets of homologous chromosomes. They have the chromosome for mom and they have the chromosome for dad. They have both of them. Now, how many chromosomes do you have in your hair cells? Okay, let's start. How many chromosomes do you have in blood cells?
46.
46. How many chromosomes do you have in skin cells?
46.
How many chromosomes do you have in hair cells?
46.
How many bone cells? 46. Okay, that's a complete set of DNA. You got half of that from your mom, you got half from your dad, you have 23 pairs of chromosomes. We would say that your diploid number is 46. Your diploid number is 46 because you have 46 chromosomes in your body cells. In your body cells. Your body cells are diploid cells. So what's a body cell? Any cell that's found in your body. What isn't a body cell? Sex cells, egg or sperm cells. So a haploid cell, or just N, is a cell that contains half the amount of chromosomes. So a haploid number is a cell that contains half the number of chromosomes. And these are sex cells, and these are sperm or eggs in humans. Okay, even like when we talk about flowers, flowers have male and female sex cells. We don't often refer to them as sperm and eggs, but they have male and female counterparts. Pollen, for example, is the male sex cell when we talk about flowers. So pollen is like sperm, essentially. Okay, so a haploid contains half of the number of chromosomes. Diploid contains both sets. So N would be like 23 for us, but 2N is 2 times 23, so 46. Our diploid number is 46, our haploid number is 23. And we'll use those terms, we're gonna use those terms kind of moving forward. Diploids and haploids. We won't use haploids yet, but we're good. Remember, we are getting a puppy named Mitosis, and our puppy Mitosis and puppy, so we're teaching her to be house trained. She needs a PMAT. The PMAT stands for prophase, metaphase, anaphase, telophase, and it's the process in which a cell goes through and divides. It's the mitosis piece. Sometimes instead of PMAT, we use the term we could use pass me a taco as a memory device. It doesn't matter. You just got to remember the order that they occur. Either PMAT or puppy mitosis needs a PMAT, or pass me a taco. That's the order in which mitosis occurs, and that's what your homework was, to kind of get you the ideas of mitosis and meiosis. We done with this? I think so, right? This is where we got everything we need?
Yep.
Okay, so what we are about to do is test. We're going to be testing in biology on December 21st in this class, which, assuming I wrote the right date, is the Monday before break. I'm telling you now, so if you know you're going to be gone on December 21st, I would recommend that you be proactive about that, and we take that prior to you not being here. Because coming back on January 4th is not probably going to be advantageous for you. Okay, on your test on December 21st, we are going to ask you to draw and label the steps of mitosis and the major events that occur in mitosis. When you get to class on Wednesday, we will ask you from memory to draw a picture of the steps of mitosis. And you aren't going to be good at it yet, but hopefully by forcing you to attempt it, it'll help you learn. So today what we're going to do is we're going to go through and we're going to draw mitosis. Okay, now when we talk about mitosis, mitosis is the PMAT. But prior to PMAT is interphase. And interphase, remember, contains G1, S, G2. And there's a sheet of paper you're going to pick up in just a little bit that Mr. DeBoer is working on right now. Just getting a little ahead. When we talk about interphase, there's 3 phases of interphase. G1, where the cell grows, S, where the cell replicates, and G2, where the cell prepares to divide. and organelles are replicated. So when we talk about interphase, there's a couple pieces of that, but what we're gonna talk about today is the PMAX. Okay, so we want you to know the major events, and we want you to know what it looks like. So we're gonna ask you to draw these cells. We are going to color code chromosomes for you. You're gonna use the same colors again, Mr. DeBoer?
Yep, red and green.
Mr. DeBoer is going to draw chromosomes in red and green. You need two colors. I don't care what colors they are. If you have pens and pencils, you can use pens and pencils. Blue pen, black pen, that's fine. We also have, okay, before you get up, I'm telling you this, so we also have colored pencils back here. If you want to have the same colors as Mr. DeBoer, you can. He's using green and red. It doesn't matter, there's two colors he's gonna use. All of these colored pencils have not either been used for an entire year, or they were disinfected between your class and this class. When we are done with them, we will put them in the dirty bucket and they will get disinfected between now and the next class. Okay? If you want colored pencils, you can take two colored pencils. They will get returned in the bucket when we're done. There's also a piece of, hang on a second. There's also the piece of paper here, or there's a copy of it over there. So you're gonna want one of these pieces of paper, whether it's here or there. If you want colored pencils, they're over there. Now, go ahead.
Red pen.
Red pen?
Yeah.
This one back here. Again, you don't have to use the same colors. You just need different colors. So how many chromosomes do you have in your skin cells?
46.
How many do you have in your hair cells? So what's your diploid number? 46. Okay, your diploid number is 46. We are not going to draw 46 chromosomes in our cell. Our cell is way too small. The diploid number of this cell is going to be 4. So how many total chromosomes will we eventually be drawing in here then? 4. Okay, we're going to be, and how many pairs of chromosomes is that then?
4. Oh. No.
4?
2.
2 pairs, right? So if we have 4 chromosomes, we're going to have 2 pairs. So when we are drawing interphase, we're going to draw a big circle in that picture, and we want to try to take the entire size. Now, this is, we're probably, maybe you're going to want to use your pencil or pen for this. If you want to use a color, that's up to you, I guess. Okay? So we're going to start with one diploid cell. Remember, a diploid is a complete set of genetic information. Complete copy of both chromosomes for mom and dad. So we have a cell that has a nucleus in it. Now we're going to draw a giant nucleus in our cell, very unproportional. But the reason is when we talk about mitosis, mitosis is making 2 new cells, and one of the key things is the division of the genetic information, which is housed in the nucleus. So we're going to draw a very oversized nucleus that takes up almost our entire cell. Okay, so we start with one diploid cell. We're gonna need, okay, yeah, you're right, Mr. I was gonna say we need another bullet point, but you already got it. Okay, so we start with one diploid cell, which has a complete set of copy, one from mom, one from dad. Okay, there's a nucleus is intact. What does a cell do during interphase?
Grow.
It grows. What else does a cell, that's G1, right? That's G1. What else does it do?
Replicate.
It replicates
DNA.
the DNA gets replicated, also the organelles get replicated, but we're focusing on the genetic information here. What else does the cell do during interphase? 90-some% of its cell's life, 95, 96, 90, somewhere in that ballpark, 90s of its life is spent in interphase, growing, copying its DNA during S2, or I'm sorry, G1 it grows, S DNA is replicated, G2 prepares for division. But what else is going on during a cell's life? If it's a muscle cell, what's a muscle cell do for most of its life?
Be a muscle.
Exactly. It
stuff.
does muscle stuff. Okay? If it's a blood cell, it does blood cell stuff. If it's a bone cell, it does bone cell stuff. It does its day-to-day function, is what we often say. Whatever that cell does, like it's designed for, specialized in, is what it does most of its life. 90-ish% of its life is spent doing this stuff. Okay, so we have a giant cell with a nucleus, and now this is where the two colors come in. We have like red squiggly line, like a bowl full of spaghetti, and then we have green, because Mr. DeBoer is using red and green to draw these, red and green. So we have a bunch of stringy bowl of spaghetti, some that's red and some that's green. Okay, so we start with key major events. We start with one diploid cell. During that cell's interphase, it's going to grow. The DNA is going to replicate, and it is going to do its day-to-day functions. That squiggly bowl full of spaghetti, we want you to know that the DNA is in chromatin form. Okay, so when we talk about DNA, our DNA is chromatin form. Not chromosome, it's in chromatin form. And chromatin is when your DNA is uncoiled. Chromatin is uncoiled. Okay, so your DNA is in chromatin form, uncoiled DNA. It's a bowl full of spaghetti. And because it's getting to be that time of year again, we have red and green bowl of spaghetti. Tastes the same.
Right.
That's just, that's
Not moldy or anything, it's just green and red. Okay,
For appetite.
so it's in chromatin form. That's interphase. G1S, G2. Cell grows, DNA is replicated, prepares for duplication. Then we get into mitosis, right? G1S, G2M phase. M phase is mitosis. Mitosis is broken into 4 parts: prophase, metaphase, anaphase, telophase. Pass me a taco, PMAC, whatever you want to remember. Key things that happen during prophase. This was our nucleus, right? Why do you think we're drawing dashed lines around it? What's happening to that nucleus?
It's growing,
Nope, not growing. It's getting chopped up, right? It's dissolving is often what we use to say. We're saying the nucleus dissolves, which means it's disappearing. It's getting chopped up. I don't know. That's what we're trying to show you, though, with that dashed line. That nucleus is dissolving. That's what that dashed line, and we're going to label that also, too, and say, like, the nucleus dissolves. Okay, so it's a key point, but we also want to label it on our picture so we can draw an arrow to it. In interphase, we had a bunch of uncoiled chromatin. Oh, during mitosis, we're going to have to split our genetic information. We're going to have to split the DNA. And our DNA in chromatin form is going to condense into something that we drew and call a chromosome. Sometimes we refer to it as chromatid, right? But chromatin condenses into something called chromosome. Then we just drew a picture of a chromosome, and it's our typical X-shaped structure. Okay, so chromatin goes to a chromatome. It's a transitional phase. Now sometimes they'll talk about like early prophase versus late prophase, and the idea is it's a progression, and depending upon when this picture was taken or drawn, it's going to look slightly different. But we are going to condense our chromatin into chromosome form. And we're going to have 4, I told you we're going to have 4 chromosomes. Now, we're going to draw 2 big chromosomes, one in red and one in green. And we're going to draw 2 little chromosomes, one in red and one in green. Now, Mr. DeBoer drew the 4 chromosomes side by side. So the big red is next to the big green. It doesn't have to be that way. There's 4 chromosomes in the dissolving nucleus somewhere in the cell. They could be in any place that they want. They don't. The big ones don't have to be next to each other. You could have a big and a little. It doesn't matter. The order in which Mr. DeBoer put them, he put the 2 reds on the left and the 2 greens on the right. It doesn't have to be that way. Okay? It doesn't have to be that way.
Why is there a bigger set of chromosomes?
Good question. So, how many pairs of chromosomes do you have?
23.
23 pairs. You got one from your mom and one from your dad. That's why we color coded these. Okay? The red came from mom. So from here on, red is mom, green is dad. Then. So we have 23 pairs of chromosomes. In our model cell, we have 4 chromosomes. We have 2 pairs of chromosomes. Okay? Now we have homologous pairs. How many homologous pairs are shown here? 2. Okay, so why are they different sizes? The reason we're drawing them different sizes is to represent our different pairs of chromosomes, or different chromosomes. Different chromosomes are different sizes, they have different genes, they have different centromere locations. They're different homologous chromosomes. So because they're different homologous chromosomes, they code for different genes along the way. Okay, so on here we have 23 pairs. Each pair codes for particular things. That's why we drew them different sizes, to show that they're different chromosomes. But the big chromosomes are homologous to one another, and the little ones are homologous to one another. So what we have, want some white out?
That's all right.
Okay. So we have a nucleus dissolves, chromatin goes to chromosome form, and then we have these little like squiggly lines coming out of the ends, and we call these squiggly things spindle fibers. And a spindle fiber is like a rope, is how I kind of like to think about it. They're a rope that is going to eventually attach to the chromosome, and it's going to pull those chromosomes around. We're going to move them to different locations within the cell, and eventually they're going to rip these chromosomes apart. They're kind of like a lasso. And those spindle fibers attach at the centromere, that little pinch point where the two sister chromatids come together. So during prophase, the nucleus dissolves. We have now chromosomes, not chromatin, we have chromosome form now. And we have some spindle fibers that are starting to form. PMAT, or pass me a taco. So prophase, metaphase, and right under metaphase, I'm going to emphasize middle, memory device, middle. So right under the word meta, we're going to want to put parentheses middle, middle. And something goes in the middle. Now all of this is about the division of what?
Cell.
The cell. And what part of the cell are we also splitting? The nucleus is gone now, it's dissolved. What are we trying to split? Besides the cell, we're trying to divide the DNA. So what do you think is going to go to the middle? The DNA, which is in what format right now? Chromosome form, right. So during middle, the chromosomes are going to go to the middle, and what causes those chromosomes to go to the middle are the spindle fibers are going to attach to the centromeres, and it's going to pull the chromosomes to the middle of the cell. Okay? The chromosomes go to the middle, and the reason is these spindle fibers are going to be pulling them. These ropes, these lassos, are going to be pulling them and tugging them to form a straight line. Middle, and these spindle fibers are going to be what attach. Mr. Bohr, I'm going to go get some water. My throat's getting really dry. I'll be right back. Just at my desk, so.
So I'm going to draw my chromosomes now across the equator of the cell, if you will. So I've got big red, and my little red.
Do they have to be on opposite sides?
No, just however you do it, we're gonna continue that same pattern. So I went red, green, green, red. And then our other task that happens here in metaphase are the spindle fibers. Attached to the centromere, and I will draw that here in a second.
Remember, the centromeres are the point at which those chromatids are pinched together. It was that protein structure, the dot, the center, where the X. Like if we were to draw an X like this, is where those two intersect. That's the centromere. But we go this. Right, so it's where the two parts would go. So we have these spindle fibers that are attaching. They don't have to be squiggly if you don't want to. A little artistic flair, Mr. DeBoer likes to add. Okay, they're ropes. They're attaching to the chromosomes. These spindle fibers are attaching at the centromeres, and because these ropes are attached, they're pulling them into position. Those are the ropes that are pulling them into position. Meta, middle, meta, middle, meta, middle. What goes in the middle? The chromosomes, the DNA. The chromosomes go to the middle. Meta, middle, meta, middle, middle. Okay. PMAC, prophase, metaphase, anaphase, or pass me a taco, pass me a anaphase. Ana apart, or you could even go with ana away. And what's dividing again? The cell and the
DNA.
DNA, which is in what form?
Chromosome.
So what gets pulled apart?
Chromosome.
The chromosomes are gonna get pulled apart. If we take a chromosome and we split it in half, what's each hand representing in our chromosomes? My hand chromosomes here. Here's my chromosome, what's this?
Isn't that
What?
chromatid?
Chromatid, right? And we call them sister chromatids, which are genetically identical to one another, right? So what gets pulled apart? The chromosomes are gonna get pulled apart, and they're gonna pull apart the sister chromatids, and we're gonna rip those chromosomes apart. So we still have our cell, the nucleus has still been dissolved. Now what's gonna happen is our chromosomes, we try to keep the order of your chromosomes the same. Okay, so Mr. DeBoer, can you show me your metaphase picture really quick? So Mr. DeBoer drew his red, big green, little red. So when we draw our anaphase, we want to try to match this. However, this is going to get ripped apart, this is going to get ripped apart, this is going to get ripped apart, and this is going to get ripped apart. Okay? So we're going to keep our big red chromosomes, but they're going to get ripped in half. Okay? Now, he drew his vertically. If you drew yours horizontally, then you would rip them apart horizontally. Okay? But that's how we want to draw it. So we're going to take that big X and we're going to rip him in half. We're going to see how we kind of splitting them. Now, you got to plan a little bit because spacing wise, but it's okay if you kind of spill up. So the big red got ripped in half. The little red got ripped in half. We're going to split the big green and the little green. Okay, just kind of try to keep that order the same. Okay? Now, what caused those things to rip apart?
Spindle fibers.
Spindle fibers, absolutely. Those ropes that were pulling that thing around are now playing tug of war. And eventually they tug so hard that they rip those things apart. Why? Because the spindle fibers are shortening. Okay, the spindle fibers are shortening up and they're pulling those things. That's gonna pull the sister chromatids apart. And we're gonna say the sister chromatids are pulled apart, not the chromosomes are pulled apart, because we're emphasizing what just got yanked apart. Now we have two sister chromatids that just got pulled apart. And what's true about sister chromatids? They are genetically identical to one another, right? So what that means is whatever genetic information is in this is also here. Whatever's here is here, here is here, here is here. So once this eventually is going to become a cell, and this is going to become a cell, but they are going to be genetically identical with a complete set of DNA. The sister chromatids pull apart, ana apart, ana apart, ana apart. Now, we're going to modify this worksheet slightly. It's not quite what we want. So we are going to take the last phase of PMAT, prophase, metaphase, anaphase, telophase, and we're going to put a slash next to it. We're going to slash it with cytokinesis. And I know the box right below it says cytokinesis. We're going to scribble that out. Okay, we're going to scribble out that cytokinesis in that next box. We're going to slash these. Now realize this is a process, it's a transition. And it's not prophase, metaphase, anaphase, telophase. It's a slow process, right? So these are getting pulled apart. The nucleus is dissolving. It's a process. It's not like a flip a switch and it's now we're prophase. Okay, there's these transition processes. So when we talk about telophase is the step in mitosis that deals with the nucleus and the genetic information. Cytokinesis is not technically part of mitosis. Cytokinesis is the pinching of the cytoplasm. We talked about it in that Pogel activity. Cyto means cell, kinesis means cutting, so it's the cutting of the cytoplasm or the cutting of the cell. Okay, so we are going to draw our cell.
Oh, shoot.
Hang on a second. Okay, so, hang on a second.
Thanks.
Yep. So, we are going to draw a peanut-shaped structure, but because of the way that we have drawn our chromosomes, we need to rotate that picture. That's not drawn
Yeah,
quite
it's one off
perfect.
my other one. Sorry about that,
I just
guys.
messed mine up.
It's okay. So because our sister chromatids got pulled apart like this, the cell is going to pinch like that. Okay, and we're working on fixing it. Okay, and we can help you work on fixing yours too. Okay, if you use a pencil, use your eraser. If you use pen, I can help white it out for you. Okay, but we want to try to draw.
At this point, I'll just take a new sheet.
What are you doing over there?
This thing is.
Blame the thing, huh?
I am blaming the thing.
It's fine before you had it. Works fine to me. Anybody need me to white out? I'm pretty good whiting out or whiter out or.
I
The
need to
wider
put it
outer,
I put mine up.
You're welcome. Anybody else?
Nope.
A pretty good whiter outer. I
I
think
think
you tried to
Mr.
do a
DeBoer needs
curve,
some help.
Mr. DeBoer.
I think
that's
Mr.
what
DeBoer
happened
needs
here.
help.
Okay.
Get out of there.
Let him try. See if he's good enough. See if he earns the
What,
title.
why does that look?
See if he earns the title.
He's pretty good.
I make a lot of mistakes.
Well, see, that's what I was gonna say. Look at you.
Gotta go straight. It doesn't work for There. All right.
Brilliant.
You can't even tell.
Yeah.
No, you can't.
All right, so because our sister chromatids pulled right to last hour, what happened is we split them vertically, like on the equator kind of thing, but we just split them differently. So it's kind of like a peanut, right? It kind of pinches around each peanut, the shell does. That's kind of what's going on. So during cytokinesis, that's the pinching of the cytoplasm. The cytoplasm is pinching back in, the cell membrane, I should call it. The cell membrane is pinching. But during telophase, the nucleus is going to be reforming. The nucleus is going to reform. So remember how we drew a dashed line to show the nucleus dissolving? Now we're going to draw a dashed line saying the nucleus is reforming. Now there's two cells, so technically it's not the nucleuses are reforming. We would say the nuclei are reforming, but let's just keep it simple and say the nucleus reforms. The nucleus reforms during telophase. The nucleus reforms. We pinch like a peanut, right? It's getting pinched. The cell membrane is pinches in, or is, yeah, cell membrane pinches. We have chromosomes, technically chromatid. We have condensed DNA that is going to start to uncoil back into chromatin. So we have chromosomes to chromatin, okay? But it's a transition process, so we're still gonna draw it in the chromatid form to try to help illustrate what's going on here. So our sister chromatids pulled apart. We have one big red line, half of the chromosome, right, a sister chromatid, in the cell on the left. We have half of the chromosome, a sister chromatid, big red line in the right. We have one big green on the left, one big green on the right. We have one little red on the left, one little red on the right. One little red on the little green on the left, one little green. We have four, right? We have four, two bigs, two littles, one of each color of each side. During this process, the cytokinesis and telophase, the chromosomes are going to uncoil. They're gonna go back to chromatin form, the bowl of spaghetti. Now we're showing you still in chromosome or chromatid format, okay? Realize it's a process, right? So they are in chromatid as a chromosome, but they will start to uncoil, and they're gonna be transitioning backwards. When we're all done with cytokinesis, we will end up with two genetically identical diploid cells. We started with one diploid cell, which means had a complete set of copies of genetic information. The diploid number of this cell was 4. The diploid number of these two cells, each cell now still has 4 chromatids. Its diploid number is still 4. Okay? So we finish up with 2 genetically identical cells. Mr. de Boer is drawing a picture from these, his bullet point cell membrane pinches to where that pinch is occurring. Okay, it's the pinch point.
So coming out of interphase into prophase,
Yeah.
are both of the, what do they call it? Sister chromatids,
Mhm.
are both of those identical?
Yes.
Okay.
Yep, because during interphase, the DNA was copied in S phase.
Gotcha.
Yep. So we are going to use this last box that we scribbled out, cytokinesis. What phase comes after telophase? Remember, it's a cycle.
After telophase?
Yeah. What comes after PMAT? Yeah, we go back to interphase. Okay? So let's label this interphase again. Because now we're back to interphase. Now, how many cells do we have? 2. The cycle is now complete. We have 2 cells. Both cells are going to have a nucleus. Okay, so we're going to redraw the nuclei back in. What form is the genetic information in? Is it chromatin, uncoiled bowl of spaghetti? Or is it still in chromatid or chromosome form where it's condensed?
Chromatin.
Chromatin, it's a bowl of spaghetti, uncoiled chromatin. Okay, so we have 2 cells that both have a nucleus, and both of them are a bowl full of spaghetti.
Spaghetti.
Okay, a bunch of uncoiled squiggly lines that we have, and we end up with 2 genetically identical, they call them daughter cells. They start with a parent cell,
Before into spaghetti.
and we end up with 2 genetically identical daughter cells. The parent cell divides into daughter cells. Now, if we were to compare, now Mr. de Boer can do this pretty conveniently, he can take his picture from interphase, which is on the front, and he can put it right below his picture of interphase that we just drew on the bottom. And what do we notice about those 2 pictures? They look identical, with the exception of what?
There's one less cell.
There's different numbers of cells, but size-wise,
What is that? Should be. Smaller.
they're smaller, right? The 2 cells are smaller than the one we started with. But what's about to happen?
It's gonna grow again.
In G1, the cell's gonna grow. And what happens to those 2 cells? They go through the cell cycle. And then they go through the cell cycle. Okay. So why does a cell go through mitosis? We're asking, why does a cell go through the cell cycle?
Because it wants more spaghetti.
What's that?
Because it wants more spaghetti.
No. Doesn't want more spaghetti. Okay. Why does a cell go through the cell cycle? Why does a cell go through mitosis, which is part of the cell cycle? Because an organism is going to grow or reproduce. We're talking reproduce asexually. Mitosis is asexual reproduction. It's in your section 3 guided notes. We'll talk about the difference between sexual and asexual reproduction in more detail. So to grow, to reproduce, to repair damaged cells and replace old cells. Okay, is why they go through this. So why does it go through mitosis, or why does it go through the cell cycle? We're really asking the same question for growth, repair, reproduce, replace. We started with one cell, one diploid cell, in the start of mitosis, and at the end, we created how many cells? 2 diploid cells that are genetically identical. But we start with 1, and we end with 2, and I gave number 3 away a little bit. Genetically, these cells are identical to one another. These cells are identical to one another. Okay, so genes and alleles, these are terms we're trying to introduce now because we're going to see it a little bit. Gene is a location on a chromosome that codes for a particular trait. Eye color is an example. Hair color is an example. Where an allele is a variation of that gene. Blue eyes, brown eyes, green eyes. Red hair, blonde hair, brown hair, black hair. Those are variations of hair color. Those are variations of a gene. Okay? So a gene is a location on a chromosome for a particular trait. Hair color is the gene where the alleles are a form of the gene or a gene. So it's a form of hair color. And what's a form of hair color that we could have? Brown hair, brown hair, red hair. Okay, those are different variations of the gene.
What are they? Blonde, B-L-O-N-D-E, yeah.
Anything you need to go back, any of my pictures or notes you need to revisit? Good?
All right, couple things I wanna talk quickly about.
16, right?
16? 16, perfect. Okay. So. Couple things for you to do as I expect you to be able to draw this on Monday, December 21st. We're gonna you are going to be asked to do this when you come to class on Wednesday, from memory. I guess this is not gonna go great for everyone, but it's a step in the direction. 2 things I want you to pick up while you're dropping off your color, assuming you use colored pencils. If you use my colored pencils, please put them in this bin on your way back. There's also 2 worksheets, and actually I will move these worksheets. Hang on, I'm still talking. Okay, there are 2 worksheets. The 2 worksheets, one is your homework for tomorrow. Section 3, meiosis, guided notes is your homework for tomorrow. On Thursday, you have focal activity on meiosis. This is a little bit longer than a typical virtual day assignment. I'm giving it to you now because of that. Okay? It's due, what is that, Monday? I think it'd be due on Monday for you guys. No, Sunday night. Okay, but that's there too. So as you're dropping off your colored pencils, if you borrowed a colored pencil, you're gonna put that in that bin. There's tomorrow's homework, virtual day homework is here. This is Thursday's virtual homework. If you wanna pick it up now, I would encourage you to pick it up. That's there. If you have questions on any of this stuff, let us know. We're kind of wrapping up, and that's a good stopping point.